A very good afternoon to all of you, my dear students. I hope you all are doing very well at your home. Okay, now let's focus on your studies. Today's topic is Cinchona. Cinchona. The synonym of cinchona is Cortex cinchonae, Countess purpurea, or Jesuit's bark cinchona. The biological source of cinchona is obtained from the dried bark of the stem or roots of Cinchona calycea or Cinchona legendriana, Cinchona officinalis, and Cinchona susiberi. All these belong to the family Rubiaceae. Then the next is cultivation and collection. This Cinchona is propagated by seed sowing method. The seeds with approximately 3 mm long and flat are picked and are used for cultivation. The seeds are sown in boxes and the seedlings are transplanted to nurseries when they reach at a height of 5 centimeter. The nurseries are covered by a roof so as to protect them from the direct sunlight. The seedlings grow in the shade till they attain the height of about 25 centimeter, and in between this period they are least transplanted twice. Cinchona grow well at an altitude of 1500 to 2000 meter above sea level. The temperature range required for its cultivation is 10 degree to 30 degree, and the annual rainfall required is 200 to 400 centimeter. When the plants are about 1.5 years old, they are transplanted to open space at a distance of 1 meter into well-drained, rich and porous soil. The plant is allowed to grow till 6 years, and then the first crop is cultivated by coppicing, uprooting or by felling method. The bark is collected till the plant is 9 years old, because after that the alkaloid content present in it get decreases. The uh, suitable season for its collection is rainy season. The trunk and the branches are beaten to lose the periderm, and the bark is removed into small pieces of 45 centimeter long and 12 centimeter in width. They are then dried under sunlight or artificial heating method, and during drying the bark attains quill shape and the color changes to red or brownish red. Then the next feature is their morphological characteristics. Depending upon the species, their color also varies. The outer surface is yellowish to brown with short fracture, and the inner surface varies in all four species. Like in Cinchona calycea, Cinchona legendriana is yellowish, and Cinchona officinalis is slightly brown, and Cinchona susibria is reddish brown. Their odor is distinguished, and its taste, it is highly bitter and astringent. Shape curved squill or double squilled. Size 30 centimeter long and 2 to 7 millimeter thick. Extra feature present in it is the outer surface consists of the longitudinal and transverse crack fissure ridges. Then the next is its microscopic character, which is most important. The transverse section of the bark shows cork composed of uniformly arranged several layers of thin walled cells containing amorphous reddish brown matter. Below cork is a radion of cortex composed of tangentially elongated parenchymatous cell with red-brown and thin walls containing small starch grain, ideoblasts containing microcrystals of calcium oxalate, which are about 2 to 6 micron long, and secretion cells are scattered in the cortex. Phloem consists of compressed and collapsed sieve tubes, phloem parenchyma, similar to cortex, and irregularly arranged large spindle-shaped lignified fibers. Medullary rays are narrow, 2 to 3 cell wide, almost straight longitudinal sections of bark show brick-shaped cells of medullary rays, longitudinally elongated cells of phloem parenchyma, and fibers with conspicuous pits. You can also see the in this figure the TS of Cinchona bark. You can draw this. It is very important. And in this figure, you can see the lichen, cork, cortex, secretion, and the actual position where all these are present in this section. Then the chemical constant. The chief identified alkaloid in cinchona are quinidine, quinine, cinchonidine. These constituents are the stereoisomers of each other. Like quinine is stereoisomer of quinidine and cinchonine is stereoisomer of cinchonidine. The other constituents available are quinarin, syncotin, hydroquinine, hydro cinconidine, cincotanic acid, etc. Quinine and quinidine has a methoxy group in it, but cinchonin and cinconidine do not have a methoxy group. Other things which are present are calcium oxalate crystals and starch grain. You can draw the structure of quinine, quinidine, cinchonin, and cinconidine from here, which are very important for exam point of view. Then last is use. It is employed as antimalarial drug, and it is also used as analgesic, antipyretic, protoplasmic, bitter stomach, and tonic. Quinidine is a cardiac depressant, and cinconidine is used as rheumatism and neuralgia. Thank you. That's all for today. Tomorrow we will come with the next topic.